Dear students, today we are going to discuss about one more important topic, that is GERL system. RL system. Already we discussed about the structure of here. G stands for Golgi complex, ER stands for endoplasmic reticulum, and L stands for lysosomes. And we already discussed the structure of Golgi complex and functions, endoplasmic reticulum structure and function, lysosome structure and functions, right? But both together, these three together called endomembrane system or GRL system, right? What this endomembrane system is? The endomembrane system is composed of the different membranes that are suspended in the cytoplasm. Here, Golgi complex, endoplasmic reticulum, lysosome, all these are organelles which are present in the cytoplasm. Already we know that, right? These are organelles which are suspended in the cytoplasm. Suspended means they are floating in the cytoplasm within the eukaryotic cell. Within the eukaryotic cell, already I told you that in prokaryotic cells, what happen? There is no nucleus, well defined nucleus, right? Here the remaining organelles are present in the cytoplasm only. But here the thing is that in your eukaryotic cell, we observe this system very well. These membrane divide the cell into the functional and structural. What happen? These membrane means Golgi complex has a membrane, and endoplasmic reticulum, lysosomes. These have this some here see membranes. I told you already, right? Here into function and structure, these plays major role. Mostly when you learn the GRL system, you come to know the functions of these three very well, right? And that's why we are mentioned here function and structurals. We come to know compartments are organelles. These membranes, actually presence of membranes, what they are doing, they are making differentiation and function and structural uh, differentiation, and that is what we come to learn these things very well. Here compartments and organelles. Organelles means what I told you, which are present inside the cell. It may be eukaryotic cell or prokaryotic cell, but eukaryotic cells are very well defined one, and they are advanced cells which are mostly present in multicellular cells like human beings and on all the animals, right? Prokaryotic cells like a bacterial cell, right? This endomembrane system consists here. First thing I already to endoplasmic reticulum, ER, Golgi apparatus means Golgi complex we can say, otherwise apparatus we can say. Why we are saying it to complex means what? It has three structures in them: cisternae, vacuoles and vesicles, lysosomes, and vacuoles and vesicles all together composed as a endomembrane system or GRL system. And we discuss how that happens and what happens in the these structures. How do they transport the food materials? How do they digest the food materials? How do they actually in the GRL system? What happens here? This Golgi complex is very nearer to the nucleus. This endoplasmic reticulum rises from here. You can observe here. This is the thing. It is the here. This total area is what? Endoplasmic reticulum. Here it is the nucleus, right? Now nucleus is right from the nucleus membrane. Double membrane structure is the nucleus. Nucleus has nuclear pore also. This nuclear from the nuclear membrane, what rises? This endoplasmic reticulum. Here these dots you are observing, na black dots. These are all the ribosomes. These are the ribosomes. Means when there is the ribosomes present on the endoplasmic reticulum, it means that is a rough endoplasmic reticulum which is responsible for the synthesis of proteins, right? Now when the proteins are synthesized here, and they are come into the cisternae of Golgi complex. Now here the protein whatever prepared, that is the primary protein, and after, by the preparation of this protein, and then that come to the Golgi complex cistern. I already, this is the area cistern. This is the here see vesicles and vacuoles are present, convex side, concave side. I told you already, right? These protein whatever entered into the Golgi complex, that will be modified, and that come to an end. Here see the swallow means what, bulged area of the cistern of Golgi complex is called what I told you, Galgian vacuole. Golgian vacuole, I told you, right? And this swollen part will be detached from this and formed as lysosome. Lysosome. What do you understand from this here? The transformation of membrane happened here. From this, from the Golgi complex, now lysosomes are formed, right? When the end part of the cistern of the Golgi complex is detached from the after the maturation, and that formed as the primary lysosome. Already two lysosomes are four types, I told you: autolysosome, primary, secondary, tertiary, right? Whatever is formed here, that is now primary lysosome, right? That primary lysosome has the hydrolytic enzymes, already I told you, and this you can come to know in the next slide very clearly. Just now I am explaining that what, how the lysosomes are formed here. Lysosomes are formed from Golgi complex, and whatever the lysosome is formed, the first lysosome is primary lysosome. Inside that has the hydrolytic enzymes, right? What happens, these hydrolytic enzymes will digest the food particles, as well digest the injured organelles which are present in the cell, as well, like mitochondria, remaining whatever the organelles may be, they will be digested if they are injured, get injured, right? And this is the function of lysosomes, I told you. And in this slide, you should know only one thing is that here it is transformation of membranes are taking place. That how that is how that happens, clearly we'll discuss in the next slide. But now you come to know endomembrane system or GRL system. Here that thing, right? What that composed with, that already I explained. First of all, one thing you should remember, immediate to the nuclear membrane, endoplasmic reticulum is present here, right? After that, Golgi complex is present, and Golgi complex has a concave side and convex side, polarity that will maintain, I told you, right? These things, and next we will discuss in the next slide clearly about it, okay? And you have to understand here these things. If you have any doubt, just text me all the things.